Bismillahirrahmanirrahim. Assalamu alaikum wa rahmatullahi wa barakatuh. Well, students, in the previous video, we basically discussed about the metabolism as well as anabolism, catabolism, and biocatalyst. Well, in this video, we are going to discuss about the enzyme, that how enzyme used to speed up the reaction. So before going on, I think so we should have to discuss about that. What do you mean by the term metabolism? The metabolism is basically the Greek word meaning change, and it was first introduced by Ibn Nafis. And Ibn Nafis stated that the first theory which ever discussed about the metabolism were by Ibn Nafis, who said that the body and its body parts are always undergoing change. I mean, there is always going on something in the body and its body part. And what was this something? Yes, that is the biochemical reaction. So, students, we all know that the enzymes are basically speeding up the reaction without utilizing their self, without bringing the change in themselves. So the enzymes are basically made up of proteins. They are not lipid, they are not carbohydrate, they are not the fats. They are what? They are the proteins which used to catalyze, or you can say that used to speed up the biochemical reaction without bringing the change in themselves. Well, the molecule at which the enzyme used to act is known as a substrate, and the enzyme convert them into the different molecule, and this is known as the product. So, to understand this, we should have to concern with the diagram. Let me to inform you that what this. Okay, if you're going to look in this diagram, then this is basically the enzyme. Enzyme is acting on this molecule. So this molecule will be known as the substrate. Wow, this enzyme is going to convert the substrate into the different molecule, and this will be known as the product. So the enzyme used to act on a molecule, and this molecule is known as a substrate, while this substrate is going to convert into the different molecule, and that will be known as a product. Hope so you get now. Now the question is that how enzyme used to speed up the reaction? Well, before going on this question that how, we should know that what is basically the activation energy. The activation energy is the minimum energy that is required by a biochemical reaction to begin. The example is very simple, just like a remote. If you're going to put one cell in your remote, in your TV control remote, it will not work. If you're going to put two cells, yes, then it is going to work. So these two cells are going to provide the minimum energy to the remote, so that remote is going to work. Similarly, in the case of the biochemical reaction, these chemical reactions are not going to begin until unless they are not going to get that energy, and this energy is going to help in the activation of the biochemical reaction and will convert into the product. So let me explain you the activation energy in the better way. Just look at this diagram. This is the stone that is from the position A to position B. We need to take this stone from position A to the position B. Well, this is the mountain. This is a hill, you can say. To bring the stone here at the top of the hill requires energy. But once the stone reaches at the top of the hill, it will easily reaches to the position B. So, similarly in the case of the biochemical reaction, this is the substrate. This substrate needs to convert into the product. But before converting into the product, that substrate need to pass through this. So it requires some amount of the energy. When we are going to provide this minimum amount of the energy, then the reaction will easily begin. So students, if you're going to look in this diagram more carefully, then a substrate will only convert into the product by the help of the activation energy. The activation energy is the minimum amount of the energy that is required to the biochemical reaction to convert the substrate into the product. So, let me explain you. The activation energy is basically the energy, the minimum energy that is required to the biochemical reaction to begin or to start. So, to start a biochemical reaction, a minimum amount of the energy is required, and that energy is known as the activation energy. So, activation energy means that activation energy is the minimum amount of the energy that is required by the biochemical reaction to begin. Now, what do you mean by this minimum amount of energy? Minimum amount of energy means, listen to me very carefully. Let's suppose there is a motor and which need to start with the energy of the 50 watts. If you are going to provide the current of the 50 watts, then this motor will run. Let's suppose if you are going to provide the current of the 30 watt, the motor will not start. If you are going to provide the current of 20 watts, the motor will not start. The reason is that the motor needs the 50 watts of the current. Similarly, if you are going to provide it 40, 30, 20, 10, 5, the motor will not begin. But if you are going to provide the energy of the current of the 50 watt, then the motor will begin. So this 50 watt were basically the minimum amount of the energy that were required by the motor to begin. Let's suppose if you are going to provide the 100 watt of the current, then you're actually providing it too much energy, right? So obviously this 50 watt current is the useless, right? So what actually I mean to say that this 50 watt will be the minimum amount of the energy. Otherwise, if you are going to reduce further energy, further current, the motor will not begin, the motor will not start. So this is the minimum amount of the energy for the motor to run. Similarly, for the chemical reaction, there is some amount of the energy which is required by the chemical reaction to begin, and this is known as the activation energy. If you understand about the activation energy, let's discuss that how enzyme is basically reducing how the enzyme is basically reducing this activation energy and how the enzyme is basically speeding up the reaction. So for that, you students, you should have to understand this thing. Listen to me very carefully. Basically, we all know that the enzyme used to speed up the reaction. How this enzyme is basically speeding up the chemical reaction? Basically, the enzyme used to speed up the reaction by reducing the activation energy. The enzyme is basically the enzyme is basically speeding up the chemical reaction, by reducing the activation energy. So what actually it means? Basically, this activation energy is acting as the barrier for the biochemical reaction. The activation energy is acting as the barrier. Just like this hill is acting the barrier for this stone. This is basically the barrier for the stone to reach from position A to position B. If the stone is not going to pass through this barrier, then stone will never ever reach to the position B. Similarly, in the case of the chemical reaction, the activation energy is going to act as the barrier. If this barrier is not going to covered by the biochemical reaction, it will never convert into the product. The substrate will never convert into the product. So that's why we are saying that this activation energy is acting as the barrier to convert the substrate into the product. So how enzyme is reducing this barrier? Basically, enzyme is speeding up the reaction to reduce this barrier. If let's suppose this is the substrate and it need to convert into the product that is a C. Now what is going to happen? This needs some amount of the energy and that will be known as the activation energy. Let me to draw the diagram. Let's suppose there is a substrate and substrate need to convert into the product. Now this activation energy is going to act as the barrier. If this barrier is not going to covered by the substrate by the help of the energy, then it will never convert into the product. So this substrate need that much energy, that level of the energy, so that they are going to convert into the product. Now what the enzyme is going to do? The enzyme is going to reduce this activation energy. The enzyme is going to reduce this activation energy. Now less time will be taken by the substrate to cover this barrier and will convert into the product. Students, did you get? Let me to explain you again. Listen to me. This is basically the substrate and this substrate need to convert into the product, let's suppose that is C. Now this substrate needs the minimum amount of the energy to convert into the product, and this minimum amount of the energy is known as the activation energy, and this activation energy is acting as the barrier. Barrier means that if the substrates are not going to have that much amount of the energy, then they will never convert into the product. Now how the enzyme is going to speed up the reaction? In the normal condition without enzyme, so obviously the substrate need more time to cover this barrier and to convert into the product. But in the case of the enzyme will reduce this activation energy. Now less time will be taken by the substrate to cover this activation energy and will convert into the product. That's why we used to say that the enzyme used to speed up the reaction. Now the question is that how the enzyme used to reduce the activation energy? Well, before going on, we should have to see through the diagram. Just check it out, this diagram. What is happening in this diagram? Basically, this is the substrate and it need to convert into the product. And this pink line is representing the activation energy. So, the substrate will convert unless it is not going to cover this activation energy. Once the substrate used to cover this barrier, then it will easily convert into the product. But the enzyme is helping to speed up the reaction by reducing this activation energy. Now the enzyme have reduced this activation energy. Now the substrate will easily cover this small barrier as compared to this large barrier. So this means that the enzyme have reduced the distance and now the substrate can easily convert into the product. Hope so you understand that. Now student, we can read from the theory as it is written here clearly. All chemical reaction requires activation energy. It is defined as the minimum energy required to start a reaction. The need for activation energy acts as the barrier to the beginning of the reaction. Enzyme lowers the barrier by decreasing the requirement of the activation energy. Thus, the presence of the enzyme, the reaction proceeds at the faster rate. So, if the enzyme is present in the reaction, then the reaction will complete in the small time. The enzyme used to enhance the speed of the reaction. Now let's move to the next question, that how enzymes lowers the activation energy. How enzyme lowers the activation energy? Well, there are some methods through which the enzymes use to lower the activation energy. One of the most important thing is that it used to alter the shape of the substrate. Let's suppose that, let me to explain you in the proper way. Well, let's suppose this is the substrate. One of the way to reduce the activation energy, the enzyme is going to change the shape of the substrate and will make it easier. So, by altering the shape of the substrate, the enzyme is basically reducing the activation energy. The next most important point is that it used to do the distribution of the charges on substrate. Distribution of charges. Let's suppose this is the substrate and the enzyme used to distribute the charges on the substrate. Let's suppose if we have the substrate having the positive or negative charge, in original it have in this way. The enzyme will distribute its charges. Let's suppose here is a negative and here is a positive, and this substrate is going to work more perfectly as compared to this one. So the enzyme basically do the distribution of the charges on the substrate, so that substrate is going to reduce its activation energy. The third and the most important thing is that the enzyme used to basically do the correct orientation of the substrate. What do you mean by the orientation? It means that it used to give a proper direction to the substrate so that it can easily join with the enzyme, and enzyme will convert the substrate into the product. Let's suppose the enzyme is like that. Sorry, not the substrate. Let's suppose the substrate is like that. Let me explain you. Let's suppose the substrate is like that. And this is the enzyme. The enzyme is going to change its orientation so that the substrate is going to convert in this direction. Now, this type of the substrate can easily bind with the enzyme. So, the enzyme is basically also reducing the activation energy by changing the orientation of the substrate. Hope so you get that. So, we have learned about the three main points which is actually helping to reduce the activation energy in the biochemical reaction through enzyme. So what these 3 points are? The first one is that it actually helps to do the proper orientation of the enzyme. Second most important thing is the distribution of the proper charges on the substrate. And the third most important thing is that it used to change the shape of the substrate. Now, you can read from here that the enzyme lower the activation energy in the several ways. They may alter the shape of the substrate and reduce the requirement of the energy for this change. Some enzymes also do by the distributing of the charge on the substrate. Enzyme may also lower the activation energy by bringing the substrate into the correct orientation to react. So, hope so you get that. Now let's see this most important point if you're going to discuss about the types of the enzyme on the basis of their existence. Basically, students, if you're going to look at here, these are basically the cells. These are, let's suppose, the cells. The enzymes used to work inside the cell. In the cytoplasm of the cell, they are known as the intracellular enzymes. The enzymes which used to work inside the cytoplasm of the cell, then they are known as the intracellular enzyme, and the best example is the glycolysis. Glycolysis, glyco means glucose, and lysis means breakdown. So, breakdown of the glucose is basically the glycolysis, and it basically used to occur by the help of the enzymes, and these enzymes are basically the intracellular enzymes, mean that the reaction is occurring in the cytoplasm of the cell, while the other cells used to work in the extracellular of the enzymes, extracellular of the cell. These enzymes used to exist in the extracellular spaces of the cell, and these enzymes are known as the extracellular enzymes, just like the pepsin. The pepsin used to break down the protein that is present in your stomach. Hope so you understand the lecture, and if anything is not clear to you, feel free to comment me so that I can try my best to overcome your problem. Best of luck, students. Listen my video again and again, watch it again and again. If anything is not clear to you, make the notes, send me the audio. Best of luck. Allah Hafiz. So, Assalamu alaikum wa rahmatullahi wa barakatuh.